problem is the market access. We are not having the access to the market. The farmers, they are not able to supply their product to the market at a handsome, you can say, rate. Then social equity is another pillar of the sustainable agriculture. We have to think over that. The food security, that means uh, every citizen of our country, and not only the country but the whole world, they must be having the staple food throughout the year and throughout their life. Then community well-being is another one, and then fair labor practices, where the labor they must be paid according to the government rules. But unfortunately, we are not able to meet out any of the three pillars. So this is overall sustainable agriculture, but our topic is on the soil microbiota, and it's a key component in the sustainable agriculture. So we will refine accordingly as we progress forward. You may ask in between as well. I don't mind if you are questioning in between, and if there is a doubt in between. So the sustainable agriculture in the climate change is coming up with the challenges. There are so many challenges where the sustainable agriculture is very hard to achieve in the climate change. So the climate change, the impact of climate change included the greenhouse gases. The main three gases, they are emerging from the agriculture sector, that is carbon dioxide, methane, and nitrous oxide. These are the three gases which are being emerged in the agriculture sector. And agriculture sector here doesn't mean the growing the crop. It also includes the fertilizer companies, fertilizer industries, then tractor and machineries which we are going to use in the agriculture. Then agriculture itself, like the emission from the paddy fields, then emission from the animal husbandry and poultry and all this. So there is an emission from the old agriculture background, you can say. Then another impact of the climate is the temperature fluctuations that is affecting the crop. Here, I'll tell you two examples. I think you have observed it or not. This time in Haryana, like where I belongs to, Hisar district. Hisar is nowadays is observing there is an increase in 5 degrees Celsius over the average temperature in the winters. So ultimately, it will affect the wheat crops. So terminal heat stress will be observed in the coming wheat crop. Last year during the winter, there was a frost or cold wave for 2 days. And actually, it was for 1.5 days only. But it leads to the necrosis of all the neem trees in Haryana. And I observed as well, unfortunately, I'm not showing the photographs here. Otherwise, I have had the photographs. And within 1.5 days, with the cold wave, all the neem trees, they got necrotic. And most of them are not able to survive in the coming season. Only few survives there. Then the climate change is always lead to the below ground microbial responses. That means there is an emergence of new microbial communities that may be the beneficial, that may be the harmful as well. And they are also triggering the responses towards the crops. Most of them are there, like emergence of the disease. New disease are coming up, new insect pests are coming up. And if we consider all three parameters like greenhouse gases, temperature fluctuation, and the below ground microbial responses, the solution lies with the microorganisms. The microorganisms till date, they were very less explored. Why I'm saying this less explored? Because till date only 1 to 2% microbial population is culturable. The rest 98 to 99% population is still lies in the soil. But it's no more culturable, and that means they are having the huge potential. So microbial interactions, it can enhance the soil health and the crop resilience. Crop resilience is the most important because right now, as I gave you the example of the wheat, So the average temperature right now at my place is more than 5 degrees Celsius. That means heat right now is facing the terminal heat stresses, and the temperature is not according to that. So what could be the solution? We can't lower down the temperature, but we can either we have to breed some new varieties which will tolerate the terminal heat stress, or we have to add or amend our soil accordingly so that the plants will be more bigger, more healthier, and having the more immune system. So that can be achieved through the microbial application. Then biofertilizers and biocontrol agents, this reduces the chemical dependency, because of all the chemicals, whenever they are synthesizing the chemicals, it requires a huge energy. Ultimately, the fossil fuels are being burned, the energy is being used, and it will lead to the global warming. Then coming to the soil microbiota, I think most of the participants or most of my fellow colleagues, they know what is soil microbiota. Actually, the soil microbiome is consists of a vast variety of the microorganisms which contribute to essential ecosystem services, including nutrient cycling, protecting soil structure, and pathogen suppression. So the microorganisms, they are performing both producing and reducing the greenhouse gases. For production, we are having the methanogens, methano plus gens. Methano means methane, and gen means generators. So they are producing the methanogen group of the microorganisms. They are producing the methane gas, and at the same time, we are having methanotrophs, methano plus trophs, methane plus trophs. Trophs means to eat. So the microorganisms which are utilizing methane as a carbon source, so they are reducing the greenhouse gases. So microorganisms are performing both the things, production as well as the reduction of the greenhouse gases. They are driving into the nutrient cycle, then they help in the plant growth and development. They are also being used, being explored. Rather, I'll not share the use, but they are explored for the biofortification of the crops with the help of the beneficial microorganisms. At the same time, the recalcitrant compounds are remediated using the microbes. So, if we say what's the role of soil microbiota in agriculture or the sustainable agriculture, the four major, you can say, things are there. Nutrient cycling is there. The nutrient cycling, it converts nutrients for the plant uptake, or you can say simply they make available the nutrients to the plant. Like nitrogen fixers are there, they are taking nitrogen from the atmosphere, and with the help of nitrogenase, they are converting into the nitrate, which is readily available for the plant. And then phosphate solubilizer is there, which are solubilizing the insoluble phosphate, which is already present in the soil. Then we are having potash mobilizing bacteria, sulfur oxidizing bacteria, then zinc solubilizing bacteria. So a lot of microorganisms are there which are helping in the nutrient cycling, ultimately making the agriculture sustainable in terms of the nutrient requirement. Then the soil structure maintenance, they are stabilizing the soil particles. The regional storms are there uh, due to the localized storms, due to the increase in the temperature. So they are stabilizing the soil particle by excreting the exopolysaccharides. They are binding the soil particles together, and they form a better soil structure and pulverize soil structure, rather you can say. Then greenhouse gas mitigation, it reduces the emission. As we discussed earlier, there are three gases which are greenhouse gases emitted from the agriculture sector, including your animal husbandry or your agriculture crops. So nitrous oxide, carbon dioxide, and methane. All these three gases, they can be maintained. You can say mitigated by using the various microorganisms. And then biocontrol, it suppresses pathogens and enhance the plant defense. Ultimately, there will be the decrease in the use of chemical insecticides and pesticides. Then, if we talk about the greenhouse gas emission, which is a major concern, the carbon dioxide contains a maximum 77% of the total greenhouse gases. its major contributor is from the fossil fuel burning. But why I'm emphasizing here? Because most of the agriculture produce or the chemicals which are being used in the agriculture, they are coming from the fuel burning by supplying the energy to the different industries for fertilizer formulation and pesticide and insecticide industries. Then nitrous oxide, it contributes 8% of the total greenhouse gases. And agriculture soils, they are the significant sources because whatever urea you are adding, it actually ammonia. Ammonia is turning into nitrous oxide. But there are many microorganisms which can convert ammonia to nitrate without converting into the nitrous oxide. So we can use those microorganisms to reduce that. Then methane is there, 15% of the total GHGs are there, emitted from the rice field and livestock are the main things. So one in livestock, we can go for the biogas plant where we can harbor the methane and then we can use as a fossil fuel, fuel. And but in the rice field, we have to use the methanotrophs, multiple methanotrophs, so that the emission can be lowered down. So agriculture's contribution. It's not only the sole crop, it includes all the practices related to the agriculture, whether it's a machinery, whether it's an insecticide or fertilizer companies, whether it's a transportation, whether it's crop fields. So around 32% of the global emission is caused by the agriculture, which includes nitrous oxide 6%, carbon dioxide 18%, and the 8% is the methane. Then fertilizer, irrigation, livestock, which are the key resources. Now, factor responsible for greenhouse gases emission, if we talk about this, I think this is a very, you can say, common thing. What are the different factors which are responsible for the GHG's emission? But let's have a look over this. Your manure, animal husbandry, rice cultivation, or the agricultural land, that is contributing to the GHGs. Fertilizers, as it requires coal, gasoline, or natural gas for their production. Then effect of even microorganisms, they are also secreting or adding up the greenhouse gas emission. In this consumption of fuel and electricity for farming or irrigation also add up emission of the carbon dioxide. Then rice cultivation, rice fields, methane emitting sources including lake, wildfires, wetland, termites, municipal solid waste, landfills in the agriculture areas, coal mines. So these are the factors which are responsible for the greenhouse gases emission, and most of the factors we can easily manage. But because of our negligence and you can see earlier the environment was not the major concern. So we can easily manage by using the microorganisms. And the effect on this, everyone knows what is the effect of the GHGs. The major effect is it elevates the overall temperature of the earth. The average temperature is increasing every year. Then destructive storms have become more intense. Then climate change, changing the water availability, like the people from the Chennai, they can understand more. The Chennai is in the red zone for the groundwater. And my own hometown, that's also under red zone because the water table goes more than 600 meters. And in some areas, we are not having the groundwater. So the changes in the climate and increasing in the extreme weather events are among the regions why the global rise in hunger and poor nutrition is there. And as per the sustainable agriculture, we have to meet out this hunger and the poor nutrition, which is our main aim, you can say, maintaining the economy and the environment side by side. So if we'll take the example of the microbial mitigation of the greenhouse gases. So microbial strategies includes, say for example, if you are aiming at the CO2 reduction, we can use this carbon sequestration, not only by the trees, like if you have gone through the carbon sequestration, most of the carbon sequestration strategies are following the agroforestry system, where the forests are, you can say, sequestering the carbon. But the microorganisms like Pseudomonas fluorescens, Bacillus mucilaginensis, and many other microorganisms. They can sequester the carbon dioxide up to some level. Then methane mitigation, methanotrophs, as I discussed earlier, there are two kinds of the methanotrophs, type 1 and type 2, depending on the concentration of the methane. They are oxidizing the methane and converting into the back into the carbon dioxide, which is less impact on the greenhouse gases. Then nitrous oxide control, we can use the nitrogen fixers rather than going for the urea and chemical fertilizers. We can use rhizobium, azospirillum, azotobacter, Klebsiella, Azolla anabina, and a number of the microorganisms are there which are nitrogen fixers. We can use mycorrhizal fungi for reducing the nitrous oxide emission, and this mycorrhizal fungi, it will mobilize the nutrient from one place to another place, and ultimately it will lower down the nitrous oxide emission. Comamex is a chemolithotrophic bacteria, which is you can say right now is very much explored because it's directly oxidizing the ammonia into the nitrate without producing nitrous oxide. Now coming to the next topic, first was the GSGs, second was the extreme temperature or the temperature fluctuations. So the microorganisms, they are already adapted to the extreme climates. As the microorganisms, they are ubiquitous in the nature. They're found in every environment, every conditions, every temperature, every pH. So we can explore their this particular character. And in case of the agriculture, there are two things which are very common. One is the cold stress and another is the heat stress. So in cold stress, the people from the Haryana and Punjab, they observed the necrosis of the neem leaves, neems last year. And this year, the heat stress is observed in the most of the guava growing farmers. So the cyclophilic plant growth promoting rhizobacteria, including Pseudomonas, Serraceae, Staphylococcus, they are producing antifreeze proteins. So we can use those. The question may arise here: the Serraceae and Staphylococcus, they are human pathogens as well. Yes, they are human pathogens as well, but they are different strains. Their soil, you can say strains, they comes under the grass status, generally recognized as safe, and all these are not the pathogens. So what's the mechanism? They are producing some cold active enzymes which improves the soil nutrient availability, and this soil nutrient availability is increasing the immunity of the plants as well as the plant health. If the plant will be bigger, it can withstand the different cold stresses. Same applies to the heat stress. Few thermophilic PGPR like Bacillus, Klebsiella, Bacterium, they enhance the plant tolerance by providing the nutrients in the extreme environment. And the mechanism is again the enzyme production and protective biofilms for the survival of the plants. Now, few adaptations are there. Few adaptations are observed in the microorganisms which make them survive under the extreme conditions. So they adapt accordingly. There is a variation in the membrane fluidity of the microorganism. They are open to have the conformational flexibility, better enzyme activity associated with the essential cellular processes whenever the temperature rise or downfall. Induction of the cold soaked proteins, which helps them to survive in the cold stress. Production of the antifreezing proteins. Excretion of high amount of exopolymeric substances with cryoprotection activity. So a number of mechanisms are there which make them, you can say, which may make them to survive under the extreme condition, extreme cold or extreme hot conditions. Now coming to the rhizospheric engineering and the biofertilizers. The rhizospheric engineering is a new, you can say, terminology. And recently, the scientists, the agriculture microbiologists, they started using the rhizospheric engineering word. And you will find many research papers after, I think, 2017-18, where the rhizospheric engineering word is being used. So the rhizospheric engineering is the same as we are doing the engineering, like in case of the software engineering or the civil engineering, mechanical engineering. But only thing is the rhizospheric engineering can be felt. It can be observed, but it can't be seen by the naked eyes. Like if you talk about the mechanical engineering, you can see the cars, trucks, they are running on the road. That's the outcome of the mechanical engineering. In case of the rhizospheric engineering, so whatever the microorganisms are there, we use a combination of the microorganisms and we engineer the rhizospheric population of the microorganism according to our requirement. Say, for example, if we have to say, combat the greenhouse gases from the paddy field, at the same time, we have to increase the productivity. We have to reduce the insect pest. So for paddy field, what we can do is we can use some sort of the anaerobic bacteria which are secreting the plant growth promoting hormones. So they will reduce the carbon dioxide emission by utilizing the carbon dioxide available in that through nutrient recycling. Then because methane is the main problem in the paddy field, so we can use the number of methanotrophs. And to reduce the chemical fertilizer, we can go with the azolla and avena combination so that we can lower down the urea application. And then we can use the Pseudomonas bacillus and other strains, including the trichoderma. We can use as a biological control. So likewise, what we are doing is we are altering the rhizospheric microflora for the betterment and to achieve our particular goal, keeping the sustainability in our mind. So the biofertilizers here, bio plus fertilizers, these are the living organisms which are providing the nutrients to the plant, and the nutrient to the plant maybe your nitrogen fixation, phosphate solubilization, potassium mobilization, and the few examples which are very well established are the Azotobacter, Rhizobium, and Frankia, Pseudomonas bacillus for the phosphate solubilization, Bacillus plasmalogenesis for the potassium mobilization. And the application method of the biofertilizers are soil inoculation, root dipping, foliar spray. Here, soil inoculation is of two types. You can add up directly into the soil, or you will go with the seed treatment here. So ultimately, we have to transfer the microorganisms into the soil, whether it's a seed inoculation or the soil inoculation. And the biofertilizers are a major, you can say, component in the rhizospheric engineering, because whatever microorganism is there in the rhizosphere, we are selecting it for a positive trait. So if we say here, this is the overall summary of the rhizospheric engineering, how we are engineering the rhizosphere. So first of all, we will see how the very first thing is what's the application, how we are doing the application and what are the results. So application can aim to three components which makes the plant microbiome, whole plant microbiome. We can go with the rhizospheric plant population, microbial population. Then the endophytic, which are residing into the roots and the shoots and the leaves of the plant. Then the phylospheric, above ground part of the plant harboring the microorganisms. So these are the three main components where which contributes the plant microbiome. And generally the agriculture practice which we are doing is the fertigation, then pesticides, crop rotation, and the biofertilizers. So these are the few practices which are commonly used by the microorganisms. But to achieve this rhizospheric engineering, the very first thing is screening of the PGP, plant growth promoting rhizobacteria. And whenever we are going for the isolation, the second step is we have to screen it for the different traits. So there are two kinds of the traits which we involve or which we aim at as a. So there is a direct mechanism as well as an indirect mechanism. So direct mechanism is directly what nutrient it is providing to the plant. So it's nitrogen fixation, solubilization of the potash, phosphate, zinc, and sulfur oxidizing. Then organic matter decomposition, like we are looking for the production of some hydrolytic enzymes which triggers the decomposition of lignocellulosic material, then hormone production like IA, gibberellic acid, then siderophore production, ACC deaminase production. The indirect mechanisms, including the production of the antibiotics, or you can say the antimicrobial compounds, which are being produced by the different plant growth promoting rhizobacteria to suppress the plant pathogens. Then siderophore comes in the both conditions, direct and as well as indirect mechanism. Then hydrolytic enzymes are there, HCN and VOCs are there, hydrogen cyanide and VOCs stands for the volatile organic compounds. Volatile organic compounds actually expressing the pathogen population. The release of the antioxidant enzymes, these are the very important because these antioxidant enzymes, this lowers down the reactive oxygen species, and the reactive oxygen species are usually they are secreted by the plant or the human being or all kind of the organism rather under the adverse conditions. Say for example, if so much cold is there, so much heat is there, the reactive oxygen species, their production is increased, and the microorganisms, they help to reduce in that by producing the antioxidants, and these antioxidants sponges the reactive oxygen species, and then osmoprotectants are being released. Now the PGPR, they belongs to bacteria and fungi, mostly bacteria and fungi. So what they are contributing is they are increasing the seed germination, they are increasing the seed length growth, they are aiming at the root and shoot growth, they are suppressing the plant diseases, and ultimately increasing the crop yield. Now for screening, we have to go with the pothouse screening, greenhouse screening, then followed by the field trials, then diversified field trials. Hello? Is somebody having the query?
No. He was
Okay,
unmuted. Yeah.
Okay, fine. So the once we select the PGPR, we go with the biofertilizer production, then application under the field conditions. This is overall how to develop, how we are developing the biofertilizers or the other plant growth promoting rhizobacteria, and what are the different impacts, and what are the different targets to be used for the selection. The application, as we already told you, the soil application is there, which includes the seed inoculation, then direct soil inoculation, and then for the plants which are which needs to be transplanted, that can be applied with the root dipping. The foliar spray is, you can say, the least, you can say, recommended one, and it has not that much impact till now, because they live on the phylosphere region, and phylosphere region is directly exposed to the sunlight, which is having the UV radiation. So their survival is very, you can say, very much concerned. Only that is deodorants. So only UV resistant microorganisms, they can survive there. Like the people, they are targeting Deinococcus radiodurans for its PGPR activity because it's resistant to the UV light. So if we'll say here, the number of microorganisms are there which ultimately benefited the plant population and their respective scientists. So I have summarized, like in 2022, we published a review article on the biofertilizers. So this is the extract of that. And nitrogen fixing bacteria, number of the nitrogen fixing bacteria are there, their impact on the plant growth, and then impact on the productivity parameters, and who reported this. Then phosphate solubilizing, potash solubilizing bacteria, and the collection of beneficial microorganisms, and the different biofertilizer, or we can say, if we are summarizing these. So the nitrogen fixing microorganisms are there. There are three categories of the nitrogen fixing bacteria: free living, associative, and symbiotic. Free living includes Azotobacter, Clostridium, Nostoc, Klebsiella. And associative, Azospilum, Acetobacter diazotrophicus. Then symbiotic includes Rhizobium, Trenchia, Anavina, Azole, and then Trichodesmicin. Then phosphate solubilizing number of the bacillus, or you can say endospore forming bacteria, they are there in the phosphate solubilizing Bacillus circulans, subtilis, then Pseudomonas strata, Pseudomonas fluorescens is also there, recently developed by the Rhizobacter industry. Then Bacillus polymixa, Bacillus megaterium, a number of Bacillus strains are there, a number of the Pseudomonas strains are there which are capable of the solubilizing the phosphate and mobilizing the phosphate. And Azotobacter and Rhizobium or Azospilum, these three are nitrogen fixers, but they have been observed for the phosphate solubilizing activity as well as. And then for fungi, we are having the arbuscular mycorrhiza, including Glomus, Vigaspora, Aculaspora, and one more indica, that is okay, forgot that. Then potash solubilizing or mobilizing, we are having Bacillus miscellaneous, Bacillus circulans, Bacillus edophicus. Then Aspergillus niger is also reported as a potash solubilizing microorganism. And Thiobacillus group of the microorganisms, they are responsible for sulfur oxidizing microorganisms, and number of the microorganisms are there which are promoting the plant growth, and they are residing in the rhizosphere of the plant, and their mechanism is vast, whether it's the nutrient and competition, whether it's you can say growth hormone secretion or triggering the SAR or ISR. Then coming to the biocontrol or the last, you can say we are going to end up very soon. So the microbial biocontrol strategies, because biocontrol is very much important, or you can say equally important to the biofertilizers. The biofertilizers, they are providing the nutrient to the plant, ultimately reducing the chemical fertilizer load. And if your chemical fertilizer load will be lower down, the overall greenhouse gases will be lower down, so we can achieve it in a sustainable manner. Second thing is biofertilizers, they are very cheaper in the price, or you can say not the cheaper, economic in case. Only a 50 to 100 ml amount is sufficient for the seed treatment for the 1 acre, and its cost is very less. In our university, we are selling it at 200 rupees per liter, but there are people in many industries in the market which are selling it at less than 150 rupees liter per liter. So this is a, you can say, economic one. It contributes a lot in the environment, and it lasts longer. It's not that the biofertilizers which you used this season will not help in the next season, because it lasts. It's a living organism. Once it established in your soil, it will last for many years. So microbial biocontrol strategies, what microorganisms they are utilizing mean is like a number of the mechanisms are there, but major are the three. There is a competition for the nutrients. There is a competition for the space in the rhizosphere. Then antibiotics, they are producing not only antifungal compounds, sorry, they are producing antimicrobial compound rather than, because it's not only the fungal phytopathogens are there, bacterial phytopathogens are also there. So they are producing some antimicrobial compounds such as trichoderma is there, streptomyces is there, and they are inducing the systemic resistance. It activates the plant defense, such as like the jasmonic acid pathway and salicylic acid pathway. It triggers ISR, and example is the Pseudomonas fluorescens. So these are the main three, you can say, major strategies which are being followed by the microorganisms. Although there are many strategies which are associated with these directly or indirectly into the rhizosphere. So if we see here the overall mechanism of the biocontrol agents. So very first, if we see here, the inoculation of the microorganism or inoculation with the biological control. So we take one by one. First is the competition for the nutrient and the space, because the root rhizosphere is a limited one. Its volume is limited, and every microorganism intend to inhabit the rhizosphere, because this is the most richest in terms of the nutrient. This is the most richest part in terms of the nutrient in the soil, because the plants, it exudates into the soil the various carbon source, nitrogen source, amino acids, and many other compounds. So the colonization is the first one. If the microorganism, if your biological control, they will colonize first, it will outcome the plant pathogens in terms of the competition for the nutrient and space. Then coming to the siderophore production, siderophore are the compound which are responsible for the chelation of the metal ions, and metal ions are the necessary for their enzymatic, you can say, activities. Mainly it chelates the iron, and it reduces, or you can say it decreases the concentration of the iron, or the limitation of the iron is there, and ultimately it lowers down the population of the plant pathogens. Then coming to the few enzymes are being secreted by the biological controls. For example, lytic enzymes are being secreted, and this will lyse the host pathogens cell wall, or pathogens overall, you can say, metabolic activities. Then antibiotics is there, antibiotic secretion is there. There are many microorganisms which are secreting the antibiotics or antimicrobial compounds, which lowers down the pathogen population. Then production of the volatile organic compound. Volatile organic compound includes your volatile organic compounds such as HCN and all these. And then the biosurfactants are there. Biosurfactant production is increasing the activity of all these chemical compounds which are being secreted as it increases it decreases the surface tension of the liquid. Then the indirect mechanism include your ISR, development of systemic acquired resistance, which includes jasmonic acid production and the ethylene production. Then phytohormone production, nitrogen fixation, and nutrient stabilization increases the vigor of the plant, healthy plant, and better growth and increased resistance towards the pathogens. And VOCs again, if pathogens attack, it will be it affects the plant growth, and the plant vigor is impacted, and ultimately it will not able to survive with the pathogens and the other environmental conditions. So these are the few mechanisms which are being used by the biological controls. Okay, biological control application and effectiveness. This is the major concern. Till date, most of the biological controls which are applied in the soil, they perform very well. But whenever we are going for the foliar spray or the foliar application, or we are targeting the insect pests which are airborne, we are having the limited effectiveness. So here I included a number of microorganisms which are there, and they are targeted organisms, mode of action. So there's n number of the microorganisms are there, their mode of action, then targeted plant pathogens, and the disease which is caused by them. So you can say agrobacterium, bacillus, and bacillus is dominating. Actually, bacillus and pseudomonas is dominating in this case. And in case of the fungi, the trichoderma is a dominating one. Trichoderma harginum, Trichoderma aspergillum, and viridi. Then in case of the Actinomyces, Streptomyces is the dominating one. So these are the few list of the microorganisms which are already reported by the various scientists for their, you can say, for biological control. These includes the rhizospheric, these are the endophytic microorganisms, and these are the phylospheric microorganisms. But as per of the best of my knowledge and my experience, we are facing a lot of problem in case of the phylospheric microorganisms. So only What we can do is we can pinpoint the phyllospheric microorganism producing some sort of the antimicrobial or anti-pest strategies, or having the potential biocontrol activity. So after that, we have to find out the active ingredient, what compound that is being secreted, and after that we harbor it, and then with the help of the chemistry people, we can synthesize it, or we can directly produce it from the microbial source, and after that we have to work on the microbial formulation. Although scientists are working on that, but very few they got succeed. And these are few research papers where they report in the phyllospheric microorganisms, which are targeting the, you can say, airborne microorganism. The major is, you can say, the Microbiota oryzae, which is responsible for the rice blast. And in Haryana as well, we don't have any chemical recommendation for the rice blast. So the bacillus species and streptomyces and calothrix, they are targeting the Microbiota oryzae. But only thing is whether these microorganisms will survive on the phylosphere of the rice or not. That's a major concern, because it's not that easy. But still, I included many phylospheric microorganisms which are targeting the different diseases. But I doubt what they are performing in the field. Then these are the microorganisms which are actually working as a plant growth promoting strains. They are by secretion of the plant growth hormones, they are affecting the plant growth. They are increasing the plant growth, and ultimately the plant vigor will save the plant itself from the different diseases and the environmental conditions. So this is how overall how we are screening the biocontrol agents and the biofertilizers. I think everyone knows that usually we are isolating the endophytes, phylospheric, and the rhizospheric microorganisms. After that, we go with the lab testing. Lab testing include your different traits, including siderophore, HCN production, phytohormone, ammonia excretion, antibiotic excretion, lytic. Once they are there, we can further go with the microbiome engineering. Microbiome engineering, that means we need to alter its genome, or by conventional practices we can increase the particular hormone, and after that we can go with the greenhouse or pothouse trials, and then the field testing. The field testing include your research farm, followed by the farmer's field, and followed by the diversified field testing. Now, I'm concluding here my topic. I think I'm well in the time, so I have to make a hurry to complete it in 1 hour, and we are having the 10 more minutes. So the key takeaways from my lecture, hopefully, the soil microbes, they play a crucial role in the sustainable agriculture. And what I can conclude here is it's not feasible for the agriculture without the soil microorganisms. And microbial-based solutions reduce dependency on the chemical inputs, including your biofertilizers and the biocontrol agents. And enhancing microbial application can combat the climate change effects, as we discussed in the management of the nitrous oxide, carbon dioxide, and the methane. And future prospect is advanced microbial consortia for targeted application. That is what I was discussing, the rhizospheric engineering. So rhizospheric engineering must be region specific and the season specific, and it must have the targeted approach. What kind of the approach we are following, like whether we have to go with the environmental, or we have to reduce the only greenhouse gases, or we have to reduce the cost of production, or we have to increase the productivity. So ultimately, we have to develop a microbial consortia which will combat all these things, all the three parameters it will meet out, including the suppression of the plant disease. Then policy support is also needed for the microbial biostimulants. By right now, like in June 2022 or 3, the government asked the different asked or you can public opinion was asked by the government for development of the biostimulant regulations. And I gone through those policy documents, and we were having only very few microbial stimulants which are included. But what I found, what our research is saying, the microorganism is having the vast, you can say, uh, vast prospects in the biostimulants. So we have to find out the different microbial biostimulants. As a agriculture microbiologist, they are not targeting the active ingredient or active compound in India. We are just growing the microorganism and then taking it to the field, but we are not going inside it. So that is much needed rather than microbial biostimulant as compared to the chemical biostimulants. Then integration of the microbial technology in precision farming is very much required, because the precision farming, or you can say the uh, controlled farming system like polyhouses and all this, they are emerging out with the new insect pest. And in our area, nematode infection is one which is a major concern. So this is all about from my side.
Thank you, Doctor. Very nice presentation was there. And I request to all the participants, please ask your questions or any query from the speaker. You can write in the inbox also. One question is there, sir.
Yeah. Dr. PK Mishra is
Right.
asking how to monitor the introduced binoculant in the plant rhizosphere under field condition. This is where you can say this is very typical, Dr. Sahab, because for monitor the introduced binoculant, what we have to do is we have to genetically mark up the microorganisms, and once we mark up the microorganisms, we can't observe it into the field conditions, because it requires some regulations from biosafety committee. So it's not recommended. Yes, however, the plot which is available for the genetic engineering, you can watch there only. We can use different tagging genes like green fluorescent protein marker is there, LAGZ is there, GUS is there, LUX is there. But till date, it's very hard, and we don't have any regulation to observe the or to monitor the binoculants in the plant rhizosphere under field conditions, because it requires once you tag the microorganism once, so that means it's a genetically engineered microorganisms. It comes under GMO rules.
Yes. So, anybody else?
We have 5 more minutes.
Yeah, 5 minutes more. So
Actually, Anju told me that they are very particular with the time, so be hurry.
Yes.
Actually, the presentation belongs to like a 1.5 hour or 2 hour
Yes.
with the discussions. But it's online. Online is having the limited discussion.
But because every assistant professor or any professor is engaged in some other work also, na? So yeah, there is limitation of time. So I think there's no questions in the inbox. There's no query because you're present. Yeah. Someone want to say something?
Yes, sir.
Again. Yeah. Yeah, Dr. P.K. Mishra.
Dr. Sharawat, it was very interesting.
Thank you, sir.
Presentation by you, and excellent presentation. I just want to ask that you are also working on mycorrhiza. Do we have any alternative for particularly the VM mycorrhizal fungi?
VM mycorrhiza, yeah.
The alternative of the mycorrhiza fungi.